Hi everyone and welcome to the lecture series of microbiology. I am Dr. Anusha Rathi. Microbiology series mein today we are going to talk about a very important topic which is bacterial genetics. It is super important from the point of view of competitive exams, frequently asked in a lot of INICTs, also important for NEET PG and MBBS second year ke liye to you cannot afford to miss this topic. Now, the questions which are asked from this topic are the ultimate understanding of it. Means they might give you situations and equations and ask you to write the final outcome of all the processes which the bacteria undertake in order to transfer genetic material. Now, we are going to take up this lecture and in a very short amount of time, we are going to understand it so that we never have to revise it again, right? So, when I talk about bacterial genetics, I mean that all the genetic procedures which the bacteria take, and what is special about it is that, like us humans, the genetic material gets transferred from parent to progeny, which is called as the vertical transmission. But these bacteria are so smart, and that is the reason that evolutionarily they have been able to outsmart us, is that they can transfer the genetic information from unrelated bacterium one to another, meaning that this is bacteria A, this is bacteria B. They are not related, means they are not parent and daughter, but they are able to transfer the genetic information between each other. Just like you and I are not related biologically, but I am able to transfer information from me to you via this medium. Okay, this is horizontal transfer of information. Genes are also information. They contain genetic codes, right? So this exchange of genetic information between unrelated bacteria renders these bacteria highly adaptable to new environment such as antibiotic resistance. That is why we see that antibiotic resistance has been spreading rampantly, because bacteria have been able to give each other the messages of how to protect themselves against these antibiotics, right? So the way in which the bacteria is able to transmit information from one to another is through four major processes. And why we are studying about these four major processes is because these are the ones which are asked in exams most commonly, and these are the ones for which you need to build the concepts, right? So we are going to cover transformation, transduction, lysogenic conversion, and conjugation, which the bacteria takes place in order to communicate with each other, such as this bacteria talking to this bacteria and vice versa, right? So when I say transformation, let's first talk about transformation. When I say transformation, it is a passive process. Okay. So, you must have seen in old movies, or also this might be the story of a lot of your parents' generation, that the people who move out of villages and move to the cities, over time, they changed completely. और जब वो गांव वापस जाते हैं तो लोग उन्हें कहते हैं तू तो पूरा बदल गया, right? Now that person sometimes is not even actively trying to change. It's just that he is in an environment where people behave in a certain manner, talk in a certain manner, dress in a certain manner. So passively that person changes. Like a lot of you might also have NRI cousins. When in India they had a certain dialect and accent, the moment they move to US or Canada, they tend to catch that accent and their accent changes. Now that is a passive process. Why is that happening? Because you're simply in an environment where things are happening in a certain manner. Similarly, these bacteria, they randomly uptake naked DNA from the surroundings and incorporate it into their own genome, right? Incorporation into their own genome. Into their own genome. That is the thing, right? So suppose this is a bacterium A, okay? This is a bacteria A, and bacteria A had its own chromosome and genetic material, right? It was just simply living its life nicely. Here was now this bacteria A, because of some reason it undergoes lysis, okay? It lyses and it breaks down completely. Now when it breaks down because of lysis, the DNA material, the genetic material leaks out from its body. Now this DNA material with its fragments is just simply existing in the surrounding nature. Now this bacteria B, here is a, let's say, bacteria B who's sitting nicely, and bacteria B in fact has its own genetic material, okay? It has its own genetic material or chromosome. But passively from the environment, this bacteria B is going to take up the genetic material and incorporate the genetic material of bacteria B into its own genome, right? So for the lack of a better color, let's use a different color here, okay? Suppose this was the genetic material. Okay, correct. So now you can see that the DNA of bacteria A has been incorporated into bacteria B passively through the process which is called as transformation. So you can see that passively the genetic material is being exchanged. Now you can't just have an hypothesis and say that things are happening like this, okay? You need to have a proof. So who proved this process of transformation? It was Griffith. Now this Griffith experiment is a commonly asked question, and instead of rote learning it and revising it again and again, once and for all, let's talk about it and finish this topic. So what did Griffith do? Griffith took a bacteria which is called as Streptococcus pneumoniae. You must have heard of this. Now Streptococcus pneumoniae, we all know, is also called as pneumococcus, right? It's also called as pneumococcus, and it's a gram-positive diplococci. So there are 2 gram-positive cocci which exist in pairs, and it has a capsule. That is the thing about strep pneumoniae. It is capsulated gram-positive diplococcus. Now, this capsulated strain is what we call as the virulent strain, means which can cause the disease. So what did he do? He took a mouse. He took a mouse and he injected the mouse with a capsulated strain of strep pneumoniae. So this is a capsulated strep pneumoniae. So we understand that a capsulated strep pneumoniae is a virulent strain, right? So he saw that the mouse obviously died. When he injected that, the mouse was killed. Then he did something. He removed the capsule. How do we remove? We do subcultures, and upon multiple subcultures, the bacterium loses the capsule naturally. So he did that and he removed the capsule. So now he has an avirulent or a non-virulent strain, which is a non-capsulated Streptococcus pneumoniae. So obviously, when there was a non-capsulated strain injected into the mouse lived. The mouse did not die, right? He lived. Then he did something else. He took the virulent strain and he applied some heat to it. So he killed a virulent strain. So what did he do? He took a heat-killed virulent or capsulated strain. So since it was killed already, the mouse was able to live. The mouse did not die. Then he did something else. He took 2 strains because of which the mouse was able to live. That is a non-capsulated avirulent strain and a heat-killed capsulated virulent strain. And he took 2 positives, mixed them together and injected into a fourth mouse. Over here, what did he do? He took a heat-killed capsulated plus he took a non-capsulated strain. Now you see, both of these are the strains which do not have the ability to kill the mouse. So the mouse must have lived. But what happened to his surprise was that the mouse died here. The mouse died. So he said that probably what is the most important thing is the presence of a live bacteria which has a capsule in order to kill the mouse. So how come over here the end product was that a live bacteria with a capsule was produced? The one which is live here is non-capsulated and the one which is dead here is capsulated. So he said that the dead capsulated bacteria gave its own capsule to the live uncapsulated bacteria. So ultimately what was produced was a live capsulated bacteria which killed the mouse. Right? So basically, there was this capsulated bacterium, which is bacteria A, and a non-capsulated bacteria B. When the capsulated bacteria A died, its DNA was taken up randomly by transformation by bacteria B, and bacteria B ended up building a capsule itself too. Right? So this was Griffith experiment. 2 types of strains, a capsulated virulent live strain, non-capsulated, non-virulent live strain and a capsulated virulent dead strain. When mixed, the virulence gets transferred from dead bacteria to a live bacteria. That was the concept of transformation proved by the Griffith experiment, right? Next, we have the concept of transduction. Transduction takes place with the help of bacteriophages, right? We know bacteriophage are viruses which phage on bacteria. Phaging means to eat. So these are viruses which love to eat bacteria, right? So these phages, they don't just eat bacteria, they also survive inside these bacteria. So what do? They transfer their own, these viruses transfer their own genetic material into a bacterium so that as the bacteria is multiplying and dividing and surviving, the genetic material of the virus also survives with them. So that's a smart way of utilizing the machinery of bacteria in order to survive itself. And not just bacteriophages, all viruses do the same thing, right? So the way in which these viruses help the bacteria in transferring the genetic material is called as transduction. Okay, there are two types of transduction that we are going to read about. One is called as a generalized transduction, wherein it is just going to generally take up some chromosome material and transfer between the two bacterium. And another is called as specialized transduction, because it is taking some specialized portion of the chromosome. So it is also called as restricted transduction, because it is only taking one particular restricted portion of the chromosome and transporting between bacteria, right? So before we go on to understand how this transduction process takes place, we need to understand how the bacteriophage attains or goes about its life cycle. So the life cycle of a bacteriophage undertakes two processes. One is called as a lytic cycle. What happens in a lytic or a lytic cycle is that ultimately the lysis of the bacteria takes place, right? So you can see over here that this is a bacteriophage. The bacteriophage has its own pink colored DNA, and the bacterium here has its own yellow colored DNA. In the next step, the bacteriophage is going to sit, we know the process, infect, penetrate and inject. So it is going to inject its own pink DNA inside the bacterium. So now you see this is the phage DNA which is pink colored. So this here is a phage DNA, bacteriophage DNA, and this here is the bacterial DNA or the bacterial chromosome. Now the phage, as the multiplication cycle takes place, the phage DNA multiplies and develops along with the bacteria's machinery, right? Then when there is time, the phage DNA, it it translates and makes proteins, and small little baby phages are produced inside the bacterium. Like the normal multiplication machinery in which the DNA material is transcribed and then it is translated into proteins. Now the phage DNA is getting transcribed and translated into phage proteins inside the bacterium using the machinery of the bacteria. So now the phage has successfully succeeded in making baby bacteriophages inside of a bacterial cell. Now these baby bacteriophages, when they're fully mature and fully formed, they have to come out of the cell. So what is happening is the first step is infection. It is infecting, means it is putting its own genetic material inside the bacterium. The next step is obviously multiplication and phage assembly, where the single bacterial, the phage chromosome has multiplied, made many phage chromosomes, translated, and one phage converted to many baby phages inside the bacterium. Now they have to release out, right? So what will they do? Pop goes the bacterium. They will burst or cause lysis of the bacterial cell and get released out of the bacterial cell. So then pop goes the bacterium. This is the final step, which is the release. This is the lytic cycle. So first of all, the phage will infect the bacterium, give its own DNA inside the bacterium. Then the DNA will utilize the machinery of the bacteria, produce many little baby phages. The baby phages will lysis or pop or kill or burst open the bacterial cell and be released outside. Now all of these phages are independent phages. They are free to go and infect another cell and continue the cycle like that. This is one way in which the phage has its life cycle, called as the lytic cycle. Then the another cycle is called as lysogenic cycle. What happens in lysogenic cycle is something even more shrewd, right? So over here, the phage becomes a part of the life of a bacterium, and the bacterium does not even realize. So it's kind of like घर का भेदी, yes. So वो उसके घर में घुस के बैठा है, उसके बच्चों के साथ बड़ा हो रहा है, और बैक्टीरिया को खबर तक नहीं है कि उसके साथ ऐसा कुछ हो रहा है, राइट? सो दिस इज बैक्टीरियोफाज. फाज आया, इन्फेक्ट किया बैक्टीरियम को, गिव्स इट्स ओन डीएनए मटेरियल इनसाइड. सो लेट्स सी that this had this pink DNA material. Now it injects its own pink DNA material, and now inside the bacterium, the green DNA of the bacteria and the pink DNA of the phage is there. Now this pink DNA will decide to incorporate and mix with the DNA of the bacterium. Instead of simply being separate and utilizing the machinery, it decides to become a part of the bacterial chromosome, right? So now you can see that it has integrated with the bacterial DNA. So over here you can see that this little portion is the phage DNA and this green portion is the bacterial DNA, and they have been incorporated together. Now as the bacteria is dividing, the phage DNA is dividing along with. So this one bacteria gives rise to two daughter bacteria, and you can see that in both these two daughter bacteria, the phage DNA is also incorporated. So that is also dividing. Now this phage will wait for its time. Here this phage was just entering, multiplying, lysing and going on with life. Here it is waiting. Now it has given its DNA to the bacteria. Now the bacteria will keep multiplying, living its life, not knowing that the phage DNA is there as a part of it. Now as the bacteria is multiplying, the phage DNA is also multiplying. Now each of these single bacteria has the ability to produce many baby phages, just like over here. So this is kind of an exponential multiplication here, right? Because it has now created multiple phage producing bacterial factories. Over here there was a single bacteria producing phage. So if you think of bacteria as a factory, then here was a single factory which could produce n number of phages. But here the number of bacteria is more. So many factories have been created which can produce that many more phages. Now it's just waiting for its time. This integration of phage DNA with the bacterial DNA, this entire setup is called as a prophage. Why is it prophage? Because this is what is going to become a phage in the future, right? And this prophage is like a ticking time bomb. You know, a time bomb doesn't burst suddenly. It waits for a time. And when the time is right, then it bursts. Similarly, here it is waiting for the right time. And when will the right time come? When there is unfavorable conditions. So whenever there is unfavorable conditions, suddenly this phage will enter into the Lytic cycle, meaning it will disintegrate, it will break off from the bacterial DNA, it will start dividing separately using the machinery of bacteria, just like it was doing over here. Now using the machinery, it will start translating into proteins, baby phages will be produced, and each one of these bacteria will lyse open and the phages will be released. So the ultimate step is lysis. Either it can straight away go into the lytic cycle, or it can take the path of lysogenic cycle and then move over to the lytic cycle, and ultimately bacteria has to lyse open and release the bacteriophages. So this is the two ways in which bacteriophage can have its life cycle. Right? Now, how does this life cycle come into play when we are talking about transduction? Let's go back to our topic. We were talking about transfer of genetic material between bacteria and how this bacteriophage is behaving like a vehicle of transport. How we transport our people in trains and planes and buses, or maybe the aliens, if they come to our earth, how will they come? Through a spaceship. All of these are what? These are vehicles of transport. Similarly, the bacteria, here the bacteria was simply, transferring its own genetic material and multiplying itself. Sometimes what happens, this bacteriophage will decide to pick up the DNA material of the bacteria and transport it to another. So first let's look at generalized transduction. I have written two words, mispacking and pick and drop service. So let's see how that is going to happen. Here you can see that this yellow bacterium is bacterium A with the red chromosome. right? The bacteriophage has come with its own pink DNA. It has inserted its pink DNA inside. Now you can see that inside the bacteria, the pink phage DNA and the red bacterial DNA is there. What will happen? The pink phage DNA and the red bacterial DNA will go into the multiplication cycle. Incorporation is not happening, prophage is not forming, right? In the multiplication cycle, the phage DNA, bacterial DNA, they will all divide, they will all multiply. Then ideally what was supposed to happen, like in the lytic cycle when the multiplication was happening, then the phage DNA should translate phage proteins and make baby phages. So the baby phages produced inside the bacteria should have the phage DNA. Yes, and then lyse the bacterium and come out. When they come out, they all have the pink phage DNA. You can see, right? But what happens sometimes? A mispacking happens. Meaning that instead of packing its own pink phage DNA, it ends up packing a fragment of the red bacterial DNA like you can see over here. By mistake, it has now packed the red bacterial DNA. And ultimately, the bacteria will be lysed and the phages released. Some of them will have the correct DNA of the bacteriophage, but some of the phages released by mistake have mispacked a portion of the bacterium A chromosome. Now, as a normal life cycle, all of these phages will go and infect new bacterium. Suppose this red DNA carrying phage goes and infects this bacteria B. So now, as a normal process, the phage will inject its DNA. But here the phage's own DNA is not the phage's DNA, it's the bacteria A DNA. So what has happened ultimately? What has ultimately happened? The DNA of bacteria A has been transported to the bacteria B. So isn't it like a pick and drop service? Essentially, if we ignore all these steps in between, which is simply nothing, which is simply the protein translation and packing of the phage DNAs. So what has happened, suppose you have a mover and packer. When you leave your hostel after MBBS and you shift at home, there are certain things which belong to you and certain things which belong to college, such as probably the bed in your room or the table and chair. You hired a packers and mover service. So they not just packed your own clothes and books, they also packed a chair which belonged to the hostel authority. Isn't that mispacking? So what have they done? They have picked up the chair which did not belong to you and moved it, mispacked it and dropped it to your house. So that is a pick and drop service. Similarly, here the bacteriophage picked up the bacterial DNA which was not supposed to pick, mispacking, and then dropped it into another bacterium B by mistake. So this is a pick and drop service. Here the phage's own DNA is not going anywhere. So that is the mistake you guys make. You guys end up saying that the phage transfers its own DNA. No, phage is simply picking up bacteria A DNA and dropping it into bacteria B. That is all that it is doing. That's the end step. So because of this mispacking, so this is like a pick and drop service. This becomes generalized transduction. There is no control over what is being transported. Randomly, some fragment has been packed and transported, right? What happens in specialized or restricted transduction? So now we are talking about specialized or restricted transduction. Over here on my left hand side, you can simply see that the lysogenic bacterium, that is which contains the prophage, is like a time bomb. So here this prophage was inside a bacterium which we call as the lysogenic bacterium because it has undertaken the lysogenic cycle, nothing else. Now let's move back to restricted transduction or specialized transduction. Here what's happening, let's see. So you have this setup. The phage has come, injected its DNA. So this pink one is the phage DNA and the yellow one is the bacterial DNA. Now you see that on the bacterial chromosome there is a red dot. This red dot is supposedly a special segment in the chromosome of the bacteria. Let's suppose that this red dot is the segment which produces the penicillinase gene. You all know that penicillinase is the enzyme which the bacteria uses to break down penicillin drugs. So the bacteria which produces penicillinase enzyme is going to be resistant to the penicillin drug, right? So this is bacterium A, which has this gene for penicillin resistance, right? Next step, what happens? The phage's DNA incorporates with the bacterial DNA and becomes a prophage. Here you can see that in the prophage, a portion of the phage DNA has been incorporated. So this has now become a prophage. Yes. Now, in the next step, when this has to happen, when it has to break off and enter into the lytic cycle, where should it break off from? The pink should break off from the green. Yes. Similarly, over here, the pink should break off from the yellow and multiply and enter into the lytic cycle. But as a mistake, what is happening? It breaks off from this point. From this point. Instead of breaking off from this point, it breaks off from this point. So this is called as mis-splicing. Because of this mis-splicing, a little portion of the bacterial DNA has also been taken along with. Yes. So ultimately what has happened? The pink should have separated from the yellow and the phage should have separated from the bacterial. But now a little bit of the bacterial, the yellow, has been taken up by the phage. So ultimately the end product which is forming is this little, the phage DNA along with little bit of bacterial DNA. And can you notice that the little bit of bacterial DNA which was picked up from the site where the penicillin gene product was there. So now what has happened? There is no mispacking happening here. Yes, because ultimately the phage is packing its own DNA only, the pink DNA. Here mispacking was happening because while only pink DNA should have taken up, it has taken red DNA by mistake. Over here, it has taken pink DNA only. That pink DNA has a little bit of the yellow DNA. That is the problem, right? Because of mis-splicing. Packing is correct, right? So now these will multiply, and as they will multiply, the little red gene will also multiply along with them. Little baby phages are produced which have their own pink DNA only with a little bit portion of the bacterial DNA. And pop goes the bacterium, phages are released, they will go and infect another bacteria B. So what will happen? The penicillinase gene from bacteria A, a restricted or specialized gene from A, will now be transported to B with this bacteriophage, right? So now a restricted or a specialized portion of the gene has been given. So pick and drop is happening in both the scenarios. Mispacking is happening in generalized transduction, and mis-splicing is happening in restricted or specialized transduction, right? I hope it was able to clear all your confusions. So we have understood both the types of transductions also. Now comes the third part, which is lysogenic conversion. What is lysogenic conversion? Nothing. The simple lysogenic life cycle which we have seen. So what is happening in the lysogenic life cycle? A part of the phage DNA is incorporating into the bacterial DNA. And at this stage, this bacteria is changed a little bit. It has become a lysogenic bacterium. This bacteria is changed a little bit. It has a little bit of bacteriophage inside it. So this type of change which is produced is utilized by bacteria in many toxin production. How? Let's see. This is a bacterium. Phage gives its pink DNA. The pink DNA incorporates into bacterial DNA and ultimately you have this prophage which has been formed, right? Now this will stay like that. The bacteria will divide by binary fission and two little lysogenic bacteria are produced. They will further divide and they will produce their own little lysogenic bacterium. So what is happening? The bacteria has been changed a little bit now. The phage is not going into the lytic cycle. It is simply continuing in the lysogenic cycle only, and the bacteria is just dividing, living its life. Right? Now suppose this pink DNA of the phage had a particular ability to produce a particular toxin. This bacteria did not have the ability to produce a toxin. The phage had the genome which codes for the toxin. Now the phage has given its own DNA to the bacteria. So it has given the ability to produce the toxin to the bacteria. Now this bacteria can produce toxin. Now the bacteria becomes toxigenic. So lysogenic conversion is a little different from transduction. Because in transduction, somehow the bacteriophage was giving the DNA of bacteria A to bacteria B. Either it was giving it totally in generalized, or it was giving a little portion of bacteria A to bacteria B in restricted or specialized transduction. But what happens in lysogenic conversion? The phage gives its own DNA to the bacterium and as a result, gives the bacterium the property to produce toxins. I hope you never get confused after this. So that is where important questions are asked. Some phage-coded toxins. What are some phage-coded toxins? Is a important, very, very important MCQ. It's a supremely important MCQ. So there's a simple mnemonic. A, B, C, D, E. A, B, C, D, E. A stands for the exotoxin A and C of streptococcal pyrogenic exotoxin. So you can remember it as SPE, A and C. SPE, A and C. Then B is botulinum toxin, C and D. So with A, B, C, D, you can also remember C, D. So you have A, B, C, D and C comes here, right? So B is botulinum toxin. C becomes cholera toxin, D becomes diphtheria toxin, and E becomes E. coli. But which toxin of E. coli? The verocytotoxin of the enterohemorrhagic E. coli, the EHEC. The verocytotoxin, which is also called as the Shiga-like toxin. So even if they give you Shiga-like toxin or enterohemorrhagic E. coli or verocytotoxin, you know they're talking about the toxin of E. coli, which is phage-coded. So A, B, C, D, E, streptococcal pyrogenic exotoxin A and C, botulinum, cholera, diphtheria and E. coli, which is EHEC. These are all the phage-coded toxins. Means these toxins which are produced by these bacteria have actually been coded by a bacteriophage. And the bacteriophage gave its own DNA to the bacterium and the bacteria converted by lysogenic conversion. This finishes 3 important concepts. So up till now, you know, transformation is random uptake by bacteria. Transduction is pick and drop service by bacteriophage. If random mispacking, generalized. If specified portion transported, specialized. Lysogenic conversion is conversion of the bacteria. Dharam parivartan of bacteria by the phage, by giving its own DNA to the bacterium. Then comes conjugation. Here now there is no bacteriophage, there is no randomness. Here now the two bacteria are talking to each other with a specialized telephone or a specialized line of connection or a specialized tunnel called as the conjugation tube or the sex pili. You all know of this. You are all aware of this, right? You have seen now, this is a beautiful image that I've picked up. You all know that these pili or fimbriae that the bacteria have, some pili are specialized because they have the ability to produce these conjugation tubes, like these tubes for intercommunication between the bacteria. So the prerequisites for conjugation is, one is this conjugation tube formation and second is plasmids. What are plasmids? You know that plasmids are these little free DNA components or small chromosomal material which is roaming about freely in the bacteria. The number of genetic code in a plasmid is 1/10 that of the main chromosome of the bacteria, right? So plasmid is freely transported between one bacterium to another. It is fastly transported also because it is a fairly small genetic code, so it can be transferred very, very fast. So the conjugation which the bacteria undertake is of various types, right? So the first conjugation is that suppose this is a bacteria, this is bacteria A, and this bacteria has the ability to produce a sex pilus or a conjugation tube, right? So here is the DNA material of bacteria A, and it has the ability to produce the sex plasmid, which is coded by this plasmid, the blue plasmid, okay, right? So since it has the ability to produce a fertility organ, we can call it the F plus bacterial cell because the sex pilus or the fertility organ is coded by it. What is this? This is the sex pilus. It has the ability to code for it, right? Now there is another bacteria, this guy, bacterium B. This bacteria does not have the ability to produce the sex pilus. It also has its own chromosome, that is fine. but that plasmid is not there and the ability to produce sex pilus is not there. So this is infertile guy. So we can call him F minus. So we have two types of cells, F plus and F minus. Now the plasmid for fertility is present in F plus cell. So it is only the F plus cell which has the ability to transfer this plasmid to this plasmid. So this guy can become a donor. So this is a donor bacteria and he's going to receive the plasmid. So this guy becomes the recipient bacteria. So what happens in conjugation is a donor bacterium gives the plasmid to a recipient bacterium. We can also call him a male bacteria and him a female bacteria. You know, just the ways of nomenclature. Now what will happen? The conjugation tube is going to get formed between the two. This is bacteria A. Yes, bacteria A will form the conjugation tube with bacteria B. While they both have their own individual chromosomes, this plasmid is going to get transferred to this guy. This transfer of the plasmid happens in a rolling circle mechanism. How does it happen? In a rolling circle mechanism. So like a circle rolls out. You know, a circle, it rolls out like a carpet unfolds, है ना? Carpet round होता है, उसको धक्का मारो तो वो ऐसे ऐसे करके खुल जाता है पूरा. Similarly, the plasmid rolls out into a circle and enters inside the second cell, right? Now, since it is rolling out and entering the second cell, then after some time, what will happen? The tube will break from the middle. After the tube has broken, what is the end result achieved? End result achieved is that this was bacteria A, this was bacteria B, right? It had its own chromosome. The plasmid which has been transferred is now present in both of them. So now this also ends up attaining the ability to produce the sex pilus. Right? So both of them have the little bit of a plasmid. So it's like a rolling circle. It does not give its property entirely. It stays in both, right? So suppose I'm your friend and we are talking through a telephone and I give you some information. Now, it's not that the information that I've given you will be lost from my brain. No. I also still have the information, but now you also have the information. So we both end up having the same information, right? So that becomes the concept of conjugation. So what started as a F plus and an F minus cell, ultimately both of them end up becoming F plus. So if they ask you the equation in exam, what is the ultimate step of this conjugation? You will write that when an F plus conjugates with an F minus, they both end up becoming F plus. This is the ultimate result. Yes, so always remember, suppose I have a little tube through which I whisper into your ear. So now it's not that the information will be lost from my mind. I will still have my information, but you will also have the information, right? Then there is a second scenario. What happens in the second scenario? Again, we have this bacteria. Okay. Bacteria has its own chromosome. Bacteria has the plasmid. Now, the plasmid falls in love with the chromosome. Plasmid falls in love with the bacterial chromosome. So, plasmid goes and incorporates inside the bacterial chromosome. Now, this is not a prophage. Phage DNA is not incorporating. Bacteria's own plasmid is incorporating with the plasmid's main chromosome. Bacteria's own plasmid is incorporating with the bacteria's main chromosome. Right? This incorporation is happening. This bacteria, with a little bit of a plasmid in itself, is able to transfer the main chromosomal genetic information at a very fast pace to others. That is why it is called as high frequency recombinant cell. Because recombination has happened. Na? We know when one genetic DNA material incorporates with another DNA material, we call that process as recombination. So here recombination has happened in the same cell, even though, but two types of genetic materials have incorporated. So it's a recombinant cell, and it's a high frequency recombinant because it is able to attach, detach very, very fast, and it is also able to transfer the genetic material very, very fast. Right? Now this high frequency recombinant cell can conjugate with an F minus cell, right? So now what will happen when this high frequency cell conjugates with an F minus cell? So here you have an F minus cell with its DNA, and here you have a high frequency recombinant cell with a little bit of a plasmid. Yes, what will happen? Conjugation tube will be formed. Now, this genetic material has to start getting transferred to this new bacterium. But now a lot more genetic material has to be transferred. A lot more has to be transferred. Plasmid was quite small. Very quickly it could be sent out. Before this transmission could be completed, the sex pilus breaks. It breaks off before entire information could be given to this person. Ultimately, what happens? The HFR remains as it is, and the new bacterial cell remains as it is. Nothing gets transferred to anybody at the end of the day. Nothing gets transferred to anybody. A little bit of the plasmid and this thing could come in, but that is so small in amount that it is not enough to transmit the entire thing. So after some time, it will just disintegrate and vanish off. So what's the end result? When an HFR conjugates with an F minus, the end result is nothing happens. Nothing happens. The status quo is maintained. HFR remains HFR, F minus remains F minus. So you can imagine HFR like a selfish guy. Okay, he's a highly fortunate fellow. He's a highly selfish guy. He does not give anything to anybody. While an F plus was quite generous, HFR is selfish. Right? Now, what this HFR formation, this conversion of a simple F plus cell into an HFR cell is reversible. You know this, you fall in love with someone and you also break up. Here the plasmid fell in love with the chromosome, they got married, and then they decided to take a divorce. So when they want a divorce, what should have happened? The blue and the red should have broken off, gone their own separate ways. But life is not that fair. So what did God decide? God decides, no beta, that is not how it's going to happen. You will break off from this point. So this green point is the new point from where they will now break off. So ultimately, what will get produced? What's the ultimate thing which is getting produced now? While this process is reversible, it does not reverse, and ultimately the red remains red, but a little bit. But the plasmid now has a little bit of red. Okay, we'll take it up again. But God decides that no, that is now how it's going to happen. You will actually break off from here, you will break off from this point only, but from here you will break off from this point. Right? So while this process should have just simply reversed, instead of simply reversing, what ends up becoming is this cell with a little bit shortened original bacterial chromosome, and the plasmid now has a little portion of the bacterial chromosome in itself. Right? So the shape of the plasmid has changed a little bit. So now we can't call it F plus, we can't even call it HFR. So we will give it a new name. This is now F prime cell. This is now a F prime cell, right? What will happen when an F prime cell conjugates with an F minus cell? Come on, let's quickly draw this. It's not a problem. So you have an F prime cell which has the ability to make the sex plasmid, and here it has its own chromosome, and here it has the little bit of a recombinant plasmid, right? It is going to conjugate with a F minus cell. Going to conjugate with an F minus cell. Here is the F minus with its own DNA. So just like the normal transmission, this is going to roll out in a rolling circle, and even if it has a little bit of bacterial chromosome, it is still a plasmid only. Small genetic material needs to be transferred. It will happen very, very fast. Ultimately, what will form? Ultimately, you will have this guy who will have received the plasmid, and you will have this guy who will still have access to its plasmid. As the conjugation tube breaks off, you can see that he has its own chromosome. He has its own chromosome. This guy still has the little bit plasmid that he has. And now this guy also ends up getting the plasmid from that guy. So now what started as an F prime into F minus will become, both will end up becoming F prime. Both will end up becoming F prime. Right? So ultimate step, what is happening here? When an F prime conjugates with an F minus, ultimately they both become F prime. They both become F prime. So now if they ask you permutation combination, it becomes very easy. F plus conjugates with F minus, both end up becoming F plus, generous guy. Selfish, high frequency recombinant with F minus, selfish, gives it nothing, nothing happens, remains the same. F prime, very identical with F minus, F plus, so both end up becoming F prime. This is the ultimate goal and solution of this knowledge that you have. This F prime conversion sometimes is also called as sexduction. Sexduction, you might find it written as sexduction in your books sometimes, right? Ultimate goal of conjugation was this. So now, to summarize, we have been able to say that transformation is the passive uptake of DNA, right? Transduction is a pick and drop service by bacteriophage, either entire thing mispacked or a little bit portion because of missplicing. Either it is lysogenic conversion, where the phage is converting the bacteria like Dharam Parivartan kind of, where it is giving its own DNA to the bacterium. And conjugation is when sex tube is formed and either plasmid is transferred or high frequency not transferred. So F plus with F minus, both F plus, HFR with F minus, nothing changes. F prime with F minus, both become F prime. So that was all in this session. Hope you have been able to build an excellent concept of the same. So these are the links to all my social media, and you can always contact me in any of these if you have any queries and doubts. I'll see you in the next class. Until then, stay in the game, stay tuned, and happy learning.